Director of Biochemical Genetics at Baylor Miraca Laboratories. Today she'll be giving a presentation on metabolomics on the screening and diagnosis of rare diseases.
Thank you. Okay. Hi, good afternoon. I'd like to thank you for the opportunity to tell you about some of the work that we've been doing in metabolomics over the past couple of years. Can you hear me? No. Like this. All right. Is that better? Okay, great. Okay. So I think we all know that the diagnosis of rare disease can really be a very challenging endeavor. If the patient doesn't fit the phenotype that we all know, then I think it's sometimes a challenge to figure out the best approach. And we know that there are significant genetic variations across the population as well as environmental impacts on those variations, and sometimes those are difficult to assess as well. And so how do you determine what the contributors are to disease and what defines disease as well? And so that's been some of the focus of our efforts over the last few years in trying to identify other methodologies that might enhance our ability to diagnose rare disorders. And so we find that there are really some significant diagnostic needs in this process. And right now, our approaches really are twofold at this point. We have global untargeted approaches to diagnosis, which would include chromosome microarrays, karyotypes, and whole exome sequencing, as well as more targeted and specific approaches that might be targeted mutation analysis. It might be biochemical screening that includes panels of analytes, that maybe includes amino acids and acylcarnitines as a standard approach, as well as organic acids, which organic acids are untargeted but are also a limited set of molecules that are typically detected, as well as a number of individual targeted tests that are like panels of analytes. And those panels of analytes are very similar, I think, to, say, NGS panels that are targeted. So that's a difference between a targeted versus an untargeted approach. You can think of targeted gene analysis and gene panels, or you can think of whole exome sequencing as the targeted versus untargeted approach, and as well as targeted approaches in panels of analytes and untargeted approaches in more global metabolic analyses. And so some of the most challenging cases that we see, of course, really involve these undifferentiated phenotypes. These might include individuals with intellectual disability, hypotonia, seizures, maybe failure to thrive, or an autism spectrum disorder phenotype. So there is really this lack of clinical specificity. And I think everybody would agree that these are very commonly identified individuals in our clinics today. And so we know to rule out disease or rule in disease that often many different diagnostic tests are required, in particular to identify metabolic disorders, but other disorders as well. And so often we need different fluid types. We might need multiple visits and multiple rounds of testing for confirmation. And this may be cost prohibitive. It may be difficult for families to get back to clinic. There may be just many really very difficult issues involved in getting this done. And to add to the fact, many disorders don't have a biochemical test that's clinically available. And so other disorders, other tests may only be available through invasive methods, which again may not be something that's maybe cost prohibitive. It may be difficult again for the family. So these are all stumbling blocks toward diagnosis. In addition, when you have VUSs that show up in either gene panel analyses or in whole exome studies, that those are often very difficult to interpret. And so you don't know if you found the diagnosis or not. And so targeted tests, importantly, are very specific. And so at that point, you really need to know what you're looking for when you're doing a targeted analysis. And so we've decided to take a much more global approach and more of an unbiased approach toward looking at disease. And I think often we're using genome testing and whole exome sequencing quite regularly now. And of course, we pair this often with phenome data, right? And you may target, in fact, your exome analyses toward the phenotype that you're seeing in your patient. And so If your patient's phenotype is slightly different than what is the published phenotype, then you may miss your diagnosis as well. And so there are different approaches, of course, that involve transcriptomics, proteomics, metabolomics. All of these processes could come into play, and most, of course, are really at the research level right now. I think we feel that transcriptome and proteome studies really are not ready yet for clinical studies and clinical diagnostic testing. However, the metabolome, we think, really has become a much more reasonable and viable alternative, because we can then do confirmation testing. Once we identify abnormalities in the metabolome, we can then do targeted testing for many analytes and confirm diagnoses. We can also do targeted gene analyses and look at specific pathway anomalies as well. So there are other ways to follow up metabolomic findings. And so, in fact, if we pair the metabolomic studies with genome data and sometimes the phenome data, we have a much more comprehensive approach to the diagnosis. And so what is the metabolome? Well, the metabolome really refers to the complete set of molecules within a biological sample, whatever that sample might be, whether it's urine, blood, CSF, or cells or tissues. Now, those molecules could typically be grouped and analyzed in different classes, whether those are small molecules or lipids or larger molecules, for example. All of our work has really focused on small molecules with lipids up to a C24. So we do definitely work only with small molecules. So the metabolome, importantly, also is modified significantly by the genome and the proteome, of course. And then that's influenced substantially by the environment and physiological status of the individual. This could be circadian influences. It could also include diet, any number of variables that can interact with metabolism. And so our rationale really has been thinking about DNA as certainly the underlying cause, but then the output and the bridge to that phenotype really is through the biochemistry. And so the biochemistry really may function very well as a real functional readout of the genome data. And so there are different methodologies to approach metabolomics analysis. And this includes, as I mentioned, extraction. So you're focusing on a certain targeted group of molecules within that extraction process. And metabolomics is typically going to be an untargeted approach, okay, versus a panel type approach. So you're going to do untargeted, and typically this is going to involve focus on certain molecular weights or molecule class, but it would be untargeted within that group of molecules. And you can use tandem mass spec, Elsea mass spec, GC, NMR, lots of different approaches. The work that we do and have been doing in collaboration with Metabolon, which is a company in Research Triangle Park, and the work that we've been doing with them is utilized UHPLC and tandem processes. And so first, what happens is we have a biochemical extraction that's a very simple protein crash, removes all proteins, isolates only small molecules, and then there are 4 injections on the instruments shown here. And then, of course, data are analyzed, looking at retention time, nominal mass, fragmentation patterns, and a QA/QC process is generated, and when we come up with basically a list of molecules that are identified in that particular patient sample. These data then undergo a statistical analysis in comparison to our population data. And our population data is an ever-growing population resource that includes patient samples as well as control samples. And Z-scores are generated in this process. And so we then interpret the metabolomic outcome based upon the Z-scores that we generate in this process. And so significant Z-scores would be Z-scores that are greater than 2 or less than -2, considering standard deviation above and below the mean. And so this has allowed us to identify new biomarkers for disease, to explore pathway anomalies across entire pathways, as well as look at the impact of various genomic variants. And so the data that we generate can be visualized in a special plugin that utilizes Cytoscape. And that's shown here. So every circle that you see is a molecule that's been identified in our analysis. And the size of the circle is relative to the degree of the z-score. Okay, and I'll show you some examples later of abnormal outputs. This illustration here is an example of a normal individual. And so you don't see any significantly elevated or reduced molecules in this analysis. Now, when we consider this pathway and we look at all of the different molecular and metabolic pathways that we could assess, as illustrated here, you can see that it's a very good coverage of most of the metabolic pathways that we know are associated with disease. And so it gives us a very good overall screen of the metabolic status of the patient at the time of the sample. And so, again, here's another example of a normal output of metabolic data. And you see that there are really only a few circles that are colored either red or blue. Red circles indicate elevations, blue circles indicate reductions, and a gray circle is within the normal range. And so this is the same pathway map shown for an individual with PKU. And so you can see that this individual has many elevations and reductions and is very different than what we would see in a typical normal individual. And if we focus in this case on just the tyrosine pathway, and importantly, this is an individual who is on what we would consider good metabolic control. Okay, and so that's important to note, that this patient is under good metabolic control and has been under good metabolic control for several years. So you can see that even though we consider the individual under good control, there is still dramatically altered metabolic pathways in this individual. And so this shows I think illustrates well the reductions that we see in some molecules and the elevations in others. The size of the circle, again, is relative to the degree of the z-score, which is, again, relative to the normal population. Now, in our validation process, which is really an ongoing process as we assess new disorders, each month we're assessing different disorders. This is just a snapshot of a few of the different disorders that we examined in our first analyses. And what's illustrated here, you've got the disorder on the left side, molecules that we know are associated with that disorder listed in the middle, and then the z-scores are shown on the right. The z-scores in blue, the circles shown in blue are from individuals who do not have that disease. The circles in red are from individuals with confirmed diagnosis of that particular disorder. And so you can see for PKU at the top that it's very clear that individuals who do not have PKU have phenylalanine levels that are within a normal range and are very, very tightly grouped. And those individuals who have PKU have significant elevations of phenylalanine. Now, if you look down at the bottom, you'll see that thymidine phosphorylase deficiency is another different kind of example where we have several molecules that may be associated with that disorder. And in fact, there is overlap between the normal population and the disease population for two of those three molecules. However, thymidine is only present and detectable in individuals with the disorder. Okay, so we don't detect that molecule in individuals without thymidine phosphorylase deficiency. And so by utilizing one or more molecules, we're able to distinguish the diagnosis in the patient. So this slide I know is very complicated, but I think it illustrates well the power of the pathway analysis in this process. So as you can see, this is the branched-chain amino acid metabolism, and I've highlighted a couple of the different disorders along this pathway. In the small graphs associated with along different points in the pathways are values associated with 40 different individuals with these disorders. And so the disorders are listed in the bottom right and are color-coded. And so if we focus first on MSUD, at the top in green, and individuals with MSUD are shown in the green columns. And you can see significant elevations of valine, isoleucine, and leucine in those individuals. And then as you go below the enzyme block, you can see that all the molecules that are below that enzyme block are significantly reduced. Okay, so it very clearly illustrates the block in that pathway. And so we can see again, if you look down at propionic acidemia, shown in red, you can see similar kinds of values where we have significant elevations in multiple molecules, including propionyl carnitine and propionyl glycine, as well as others. And then you see reductions in 3-methyl adenosylcarnitine after the block. Okay, so then if we look again at a specific individual with maple syrup urine disease, again, this is an individual who we would consider under reasonably good control. And you can see that their pathway has many different alterations. And again, if we look specifically at the branched chain amino acid pathway, which is represented here, you can see these significant elevations in the branched chain amino acids across the top in red, and then the molecules below that enzyme block are in blue. And those are all significantly reduced. And so we can also, while this is a very simple example, I think it illustrates well the possibility of developing disease scores or pathway scores relative to risk for certain disease. Okay, and while again I think PKU and MSUD are very, very straightforward, we can use this approach for other disorders that may be less clear. So I'd like to spend the rest of the time telling you about two cases that I think really benefited and show the benefit of using metabolomics screening approach for diagnosis of a rare disease. And so the first case shown here is a 19-month-old male with global developmental delay and hypotonia, had abnormal movements, abnormal MRI with delayed myelination, but did not have seizures. Okay, and he had had a normal prior workup that included chromosome microarray and more or less a standard metabolic workup. And so exome sequencing and metabolomics were ordered at the same time. And exome sequencing in our facility takes about 3 months. The metabolomics takes about 3 weeks to turn around. And in this individual, we had I'm going to tell you first about the whole exome sequencing results, but the metabolomic results came out first. And they were intriguing to us, and we at the time didn't quite fully understand them. But when the whole exome data came out, we were able to pull everything together and make sense of all of the data at the same time. And so this individual had autosomal recessive genes identified with a pathogenic mutation in URAC1, which is associated with urocanase deficiency, and 2 heterozygous VUSs in ABAT, which is associated with GABA transaminase deficiency. He also had several single heterozygous VUSs. And so the focus really was on these 2 autosomal recessive disorders. And the first being this URAC1, where a single heterozygous pathogenic variant was identified. And so you always have the risk that even though you think you may have good coverage, that there may be a deletion on one allele that might be coming from one parent. And so that's always the question at play. And then the 2 heterozygous VUSs, again, are VUSs. These were missense mutations, and while they were inherited in trans and predicted to be deleterious, we did not have any confirmation to know that they were actually causative. So our focus really were these 2, and we thought that metabolomics data actually could help us analyze these pathways. And so when we first looked closely at the data, we found that there really were no abnormalities in the histidine pathway. Urocanase is involved in the degradation of urocanic acid, and typically we would see elevations of imidazole, propionic acid, as well as urocanic acid in these cases, and we saw no elevations at all in this pathway. And so this pathway was not altered. However, we did identify this molecule called 2-pyrrolidinone. And we did 2 different individual samples on this patient and got very similar results with the elevation of this molecule. And it turns out that 2-pyrrolidinone is a stable cyclized lactam form of GABA. Okay, so this would be indicative of elevations of GABA in the plasma. And so we also, you'll notice here, I wanted to point out that you'll see that there are several cases where there are elevated values of 2-pyrrolidinone in cases in individuals who have Z-scores above 2, so we would consider those significantly elevated, but these individuals did not have this disorder, okay? And it turns out these individuals were all taking either vigabatrin or topiramate, which have both been shown to cause elevations in 2-pyrrolidinone. And so if we look at GABA transaminase deficiency, as shown here, GABA transaminase converts GABA to succinic semialdehyde. And so when it's deficient, you get elevations in GABA, and then the elevations in GABA then are converted to, GABA is then converted to 2-pyrrolidinone. Okay? And so this is consistent with these elevations that we saw in this individual. And we were unaware of this particular biomarker. This is not necessarily a new biomarker. It has been published, but it's was not one that we were aware of for sure. And so it was a very interesting educational process for us. And the fact that we could identify this in plasma was new. Okay, so GABA levels, of course, are typically assessed in CSF, and that's a special test you have to ask for, and it's not something that's done on normal neurotransmitter analysis. So this was a nice finding because now we can analyze this, and we can look for this disorder within plasma. And so GABA transaminase is a rare disorder. Only 3 families have been reported with similar phenotype to the patient that we identified here, except that seizures are typically refractory. And so this patient did not have seizures at the time he was evaluated. He does have seizures now. And so again, CSF neurotransmitter analysis with elevated GABA would be the typical approach to diagnosis. And so here we've been able to use the metabolomics to rule out an abnormality in one pathway and confirm that abnormality in another pathway. And so this was a very productive approach for us. The second case that I'd like to present, again, I think you'll see is very similar in that the child has global developmental delay with increased tone, no seizures, otherwise normal growth, with a significant prior workup that was all normal. That included a standard metabolic workup plus additional procedures as well, including CSF analysis. And so metabolomics was ordered. This child did not get whole exome sequencing. And so the metabolomics profile showed an elevation of succinyladenosine. Okay? And so we then followed up this elevation with targeted analyses in urine and the CSF that was available and confirmed that diagnosis of ADSL deficiency. And so we were then able to do targeted analyses in this child and look at just the ADSL gene and identify 2 VUSs. And so this was a very rapid approach to diagnosis for this individual. And again, associated VUSs that are now known to be pathogenic with this disorder. And so adenylosuccinate lyase deficiency is a rare disorder, fewer than 100 cases reported, typically with encephalopathy, significant developmental delay and intellectual disability, along with autistic behaviors, and almost 100% of the time seizures in an infantile presentation. And so the gene is ADSL, and diagnostic features typically, diagnosis typically involves looking for the presence of SICAR and succinyladenosine in body fluids. And so we do offer a purine panel in our laboratory, and so that would be the typical approach to diagnosis would be to request a urine purine panel. However, this child did not have seizures, and so this group of disorders was really not under consideration by the clinicians. And so this is just to illustrate the pathway that's impacted here, and you typically see elevations of SICAR and acetylcysteine in the two I don't think I can show you, in the two areas where, we would see these elevations. Our metabolomics process does not identify SICAR, so we were not able to identify that molecule. We were only able to identify the succinyladenosine. And so this is an illustration from the cytoscape pathway showing this significant elevation of the N6-succinyladenosine with other molecules in the pathway normal, which was consistent with the purine analyses that we also did. And so this illustrates this single individual from the metabolomics analysis compared to the rest of the population that we've assessed in this study. But since that time, so this was within about a 6 to 8 month period, we identified 2 other families that were impacted by this disorder. And this has allowed us to expand the phenotypic spectrum of this disorder, as well as really think about utilizing this approach for rapid screening of mild phenotypes. And so in the first family that I just discussed is shown here with these 2 VUSs. There was a second consanguineous family that had a known mutation. And the third family is actually quite interesting, shown here. This family has 3 affected, or 3 boys with various levels, degrees of intellectual disability. The oldest child has only intellectual disability. The middle child has intellectual disability, seizures, and oculomotor apraxia. And then the youngest child has only developmental delay. Okay, and so the approach in the clinic was to focus on the middle child who was most severely affected. And they ordered whole exome sequencing and metabolomics off the bat. This was the first set of tests that they did. And we, as I mentioned, the metabolomics comes back in about 3 weeks. Well, when the significant elevations of succinyladenosine were identified, we knew right away this had to be the diagnosis, and we obtained samples from the other siblings as well. And we found that the youngest child also has the same diagnosis, and the older child does not have this disorder. Okay, his urine purine analysis was completely normal. And so we'd hoped that maybe we could circumvent the whole exome process and do targeted analyses. Unfortunately, the process was in full go at that point, and so we weren't able to come back on that exome analysis. So when the exome data did come out, the individuals had a known mutation in this gene, and so this was a really good example that we could utilize the screening approach and then follow with targeted analyses. Now, for the other individuals in the family, it was a very rapid approach to diagnosis, certainly for the youngest child. And so this just illustrates the urine purine analyses that we did in families 1 and 3, showing significant elevations in the affected individuals and then normal values for the one child. And so using this process, we've confirmed diagnoses in a number of different disorders and ruled out diagnoses as well in a variety of different patients in collaboration with whole exome data. And so this has been a very fruitful approach to diagnosis of rare disease. And so I'd like to think about utilizing clinical metabolomics as an initial screening tool. That's something that we've started to do in our clinics, and it's been very productive, and you get a very quick response. I think we know that the pickups on acylcarnitine and plasma amino acids and urine organic acids are very low. So far, our pickup in this process has been about 8%. So 8% of the samples that we have analyzed have either have typically confirmed diagnosis in individuals with disease. And so when we pair this process with, again, with genomic data, it's become very powerful. Certainly I've shown you some examples where we've been able to use the screening as really the primary approach, but paired together with genomic data really helps to inform the genomic data that you see from whole exome or panel targeted studies. And so this work was done in collaboration with Metabolon, as well as a number of individuals at Baylor. And so those individuals are shown here. Thank you.
Sarah, that's fascinating. Could you say a word or two about the nuts and bolts of it? What samples are you submitting, and what methodologies are you doing in your overall?
Yeah, so we're looking, plasma has been our primary focus, and this is plasma from an EDTA tube. So that's been the best approach. Heparin can be an interfering factor, and so we've avoided heparin. Some of our initial studies utilized heparin, but EDTA has been a much better source in plasma. Now, it's important that the plasma be handled properly, and the initial blood sample be handled properly, because if the sample has a delayed separation of plasma, what we find is that there are many metabolites that are either elevated or reduced because those cells are still metabolizing. So that's a significant concern. And so we always recommend having plasma spun down and isolated within 30 to 60 minutes max for the best sample. Okay, and in particular, that's going to impact some, of course, some amino acids, but TCA cycle intermediates in particular. So we're also using urine as well. I think urine is a good source, but because of the concentration differences in urine, I think it's not as reliable for diagnosing all disorders. I think that for some disorders it's been very good. So we had an interesting case of a complicated tyrosinemia where we were able to identify 5-aminolevulinic acid in an individual with tyrosinemia, whereas who we had plasma and urine samples for. Plasma was completely normal, so the urine was very helpful in that case. So you have to really think about the disorders that you're thinking of for sure. Creatine transporter defect would also be another example for that. CSF, we're also using CSF. So, good experience with that, but more experience with plasma at this point. Okay, it's a simple extraction. It's basically a protein crash to isolate small molecules, and so 1,500 Daltons is our maximum size, and this is all UPLC mass spec and completely untargeted approach. It's really dependent on our library. The library has about 4,500 molecules, 2,500 human specific. We also do see bacterial molecules as well and drug metabolites.
Sarah, that was a really excellent talk, very elucidating about how helpful it can be. So we're currently in the era where costs are really important, and 90% of the patients that are undiagnosed showing up to neurologists, for example, seizures, developmental delay, plus minus autism.
Yes.
So this, the question is, $6,500 for exome sequencing, et cetera. So from a practical point of view, outside of the academic centers interested in accumulating research data, how expensive is this right now? How likely is it to become generalized availability in the next 2 years? Those sorts of things. What are some of the considerations for that?
We are charging, we do offer this clinically, and so we are charging $1,000.
$1,000?
And so it's quite reasonable from a cost perspective. If you look at plasma amino acids, acylcarnitines, and urine organic acids, you're going to be pretty close to that price. So this is very cost effective. When you look at adding in the purine and pyrimidine panels and neurotransmitters and other molecules, those costs typically, they're going to add up to about $4,000 in total cost. So it is a cost-effective approach, for sure. It's an expensive approach right now. So will costs go down? I hope so. I hope that we can do it less expensively. But right now, this is the minimum that we can do at this point.
Well, think of it as an equivalent to an MRI cost.
Yes, that's right. No, actually,
So we
yes.
can make a very effective argument to the neurologist
It
about
does.
this.
When you look at the patients that we've assessed, and most of our actual pickups have come from the neurogeneticists that we have at Baylor, and it's been very productive. And when you look at the number of spinal taps that have been done and the number of MRIs, this was a very cost-effective approach, no doubt about it.
You said plasma amino acids, ACE, What would, for the three, when a physician orders a metabolic panel for a lab, what would that be
cost?
more than $1,000
It's
typically?
yeah. I mean, it's going to be right around $1,000, right? I think there's lots of shaking heads around here. So yes. Yeah. Yes.
Hi, very interesting. I have a question about your reference values. How do you achieve, or what's the size of your reference population, and what kind of variability do you see if you are constantly updating it, since your Z-scores are based on that? So if you could just comment on that, that'd be great.
Yeah, it's a good question. So our normal ranges are based on, like I think anybody's pediatric diagnostic biochem lab is based on the samples that you receive, right? And so that's part of it. We do have some normal controls, a subset of normal controls that are identified across general population controls. This population right now these are random samples, so these are not fasted because of the difficulty in getting any kind of fasted sample, pediatric fasted samples. So these are random samples, so they're typically fed, in that state. I hope that at some point we may be able to distinguish and separate those cases from fasted and non-fasted, but we're not to that point yet with our population. We do, let me think about how to explain this. So we have some normal controls. We have the normal population. So you can imagine that still most samples we receive are normal, right? As in most biochemical testing, most samples we receive are normal. And those individuals who have normal results then go into that general population data pool, okay? That becomes part of our reference-based population. Okay, so the abnormal cases, the ones with diagnoses, are removed and pulled from that population. Okay, does that help?
Size-wise, just
Size-wise.
approximately.
Yeah, size-wise. I think we are up to now around 800 individuals in that population. And these are pediatric cases.
Great, thank you.
Yeah. Yes.
In terms of sample preparation, because sometimes when you do lipid extractions and if you analyze lipids,
yeah.
and how do you deal with, because you want to enrich, obviously, the detection and increase sensitivity. So in terms of, do you have to do 2 different types or 3 different types of sample preparation?
So we, to keep costs down and reduce costs, we do not do multiple preparations, that's for sure. It's important to remember, I think, like with any biochemical testing, that what you're getting is a snapshot in time, and so that's important. We do see good reproducibility across samples and across days. As you know, with mass spec analyses, there can be batch effects and things like that, so we do see some impact of that. And have a good handle on how to analyze those kinds of data. For lipids, we do see lipids in this analysis, but it's limited by size. So we don't see anything larger than C24, and but up to there we see a lot of lipids. And I think the challenge with lipids is that there's not a lot of data on them. And there are many, many lipids, and most of what we see really is associated with diet, and so that's really challenging to separate that. So I feel like that the data are there, but we need better understanding of all of these different lipids that we see. So that's probably been the greatest challenge.
So
Yes.
thank you very much. It's like amazing technology to hear about, especially for someone like me that lives outside of this world. I just wanted to have you help me clarify one thing. Towards the end, you were talking about using it as an adjunct to newborn screening, but you're really talking about for children that have already had an abnormal screen, not as a primary newborn
Not
screen.
necessarily. So, yeah, so it's a good question. I think really where I was thinking was not as a follow-up to newborn screening. I think right now our turnaround time, I think, is too long for that. I think that you need to do the targeted tests that are appropriate for the follow-up to newborn screening. But I think this is for the kids that had a normal newborn screening.
But who already have symptoms of
Who have something else going on. Yes,
right.
obviously,
'Cause
yes.
The only reason I want to clarify that, because that wouldn't really count as screening, because with screening it's...
So no, I agree with you. So no, I mean, I think we would ultimately propose that this become, that some kind of approach like this become a supplement to newborn screening.
All right.
And because these kids, a lot of them just have developmental delay, and then they're not brought in until they've got obvious developmental delay at age 2 or 3 at the earliest.
So you see like a time when it would just become part of a routine test that everyone would get.
Yeah.
So that brings, it's always helpful when I hear these kinds of things and people pushing towards universal adoption, and then with your comments about cost effectiveness, what's your relationship to Metabolon?
To Metabolon. It's really started, it's an academic collaboration. That's really been our collaboration with them.
Mm-hmm.
Yeah.
But so they're funding some of this stuff too?
No, they aren't actually funding this. So they are performing the test for us, which is something that we've worked with them to develop. But it is a we pay them. It's a fee for service process.
Okay.
Yeah. So it is a fee for service.
Okay, that's helpful.
Yeah. Mm-hmm.
Everybody else answered all my long list
Okay.
of questions, so I'm down to sort of my picky things. What about like incidental or secondary findings? Like, do you have transitions in there for, say, marijuana use, cocaine use, things like that? And when you see those, how do you deal with it? And then have you come across any cases that have led to like novel treatments that you're working?
Yeah, it's interesting. You know, we have yet to see illicit drugs identified. I'm sure it's going to happen at some point. We initially had proposed to just blind ourselves to those molecules so that we would not see them, and we have not done that yet, but we'll see once we identify the first case. And so I think that's a challenge, you know, what to do with that. I mean, I think it's maybe not so different, you know, if when we're looking at pediatric samples to identify something like that, it's probably not so different than to look at a chromosome microarray that has significant consequences toward thinking of incest, right? You know, you can think of these things where it's your duty to report. So that's, you know, I think that's, you know, part of our feeling is that we need to know about those cases and report those as well. So I think we haven't seen it yet, but we can, and we know we probably will at some point once our population gets large enough. So.
Okay. Thank you very much.
Yeah, thank you.
What a way to end.